Good morning, everybody. Welcome to the November version of IIDR Joint Rounds. I'm Lori Burrows. I'm the Associate Director of the IIDR. And it's my pleasure today to welcome Dr. Sarah Kahn and Allison Guitor, who is going to give us a talk on their work on glove-based care in the NICU. So I'm just going to give you some brief bios first. So Dr. Kahn's going to speak first. She is a pediatric infectious disease specialist at McMaster Children's Hospital and an assistant professor in the Department of Pediatrics at McMaster. She's the Associate Medical Director for Infection Prevention and Control at Hamilton Health Sciences, and her research and academic foci include infection prevention, antimicrobial stewardship, and more. Today, she'll be speaking to us about preventing infections in the NICU using glove-based care and the impact of probiotics. And our second speaker today is Allison Guitor. She's a PhD student in Jerry Wright's lab. Her thesis has focused on developing a targeted DNA sequencing method to detect antibiotic resistance genes. And she's interested in how various exposures, including to antibiotics and probiotics, impact the diversity of bacteria and antibiotic resistance in the human gut microbiome. And today she'll be speaking to us about preventing AMR in preterm infants using probiotics. So thank you both for speaking. And I think Sarah's going first, and we'll hold the questions to the end. So if you have questions, please type them in the Q&A box and we'll address them at the end of the session. Thanks.
Thanks for that intro, Lori. So as Lori mentioned, I'll be going first, and the title of our talk is Preventing Infections and Antimicrobial Resistance in the NICU and Beyond, considering gloves, probiotics, and looking at poop and beyond. So with that, we'll be speaking a little bit initially about what is late onset infection in the NICU and what evidence do we have for the use of non-sterile gloves. We'll speak again about our glove care pilot study and some of the newer results we have and next steps. Then I'll shift gears and speak a little bit about probiotics and NEC and hand over to Allison to talk about the neonatal resistome and specifically the AMR bait capture technology at the IIDR. So just again, to ground us, late onset infection is a main predictor for neurologic sequelae in infants that have been admitted to the NICU. And for the large part, it is preventable infections. Most concerningly are when we have sterile site infections, so bacteremia or bloodstream infection, central nervous system or meningitis, or urinary tract infections. And much of this results in preventable complications, including increasing costs, longer NICU stays, and of course, long-term sequelae for these infants. Of course, NICU babies have multiple risk factors for late onset infection. Those include their immaturity, their low birth weight, and a variety of factors, including their immature immune systems, fragile skin and mucous membranes. A lot of the interventions we do to them, including parenteral nutrition, surgery, having to intervene for respiratory or cardiac diseases, and congenital abnormalities. One of the foci of this talk will be the discussion around handling by healthcare providers and how hand hygiene or potential barriers could further reduce this risk for infection. So what do we know, or what evidence do we have to prevent late onset infection in the NICU? So standard of care includes environmental cleaning, equipment disinfection, including isolates, breast pumps, various equipment used in the NICU. We also have infection prevention bundles, things specifically developed for catheter-associated bloodstream infection, urinary tract infection, and ventilator-associated pneumonia. We also do antimicrobial resistant organism surveillance. So we look for and try to eliminate transmission of things like MRSA and VRE. Of course, we try our best to minimize invasive procedures, but tubes, lines, and catheters often need to go in for the care of the infant. Hand hygiene is the sort of mainstay, the crux to prevention of infection. But what about non-sterile gloves as an added barrier? So this has been looked at in adult and pediatric ICUs and wards, and there's been mixed results. So there has been a study based out of McMaster that looked at universal gloving and gowning in adult ICUs, and there was a lower risk of MRSA acquisition in ICUs. Also, we know that isolation of C. difficile colonized adults prevents hospital-acquired C. diff infection and transmission to other patients. From pediatric data, there's been studies looking at universal gloving and gowning in the PICU post-solid organ transplant, and there was a reduction in nosocomial infection. Similarly, acute care inpatient pediatric units that did mandatory gloving showed a reduction in central line-associated bloodstream infection as well as hospital-acquired pneumonia. Now, you can imagine the adult and even the pediatric patient is a little bit different than a neonate in terms of sort of the degree of lines, tubes, and sort of where they're situated across the body, but also how our hands-on contact is very different for a NICU patient compared to an adult or a pediatric patient. Hence why we may see different results. This is a busy slide, but I kind of want to walk you through how we talk about hand hygiene in the hospital. And so this sort of figure is taken from the PIDAC guidelines specific for the NICU. And the expectation is that when people enter the hospital, that they perform hand hygiene sort of at the entrances. Then once you get to our NICU, for example, you're expected to actually perform a scrub all the way to the elbows and be bare below the elbows as you enter the NICU environment. Once you get into sort of the bedside area, that immediate care environment, we start to call this moment 1A. And that's, again, where we expect hand hygiene or clean hands to occur. And then once we get to 1B, that's when you're going into the isolette. And that's sort of the most critical point of contact where, again, hand hygiene is recommended. Now, the premise behind what we wanted to look at is, will adding gloves at this stage be another barrier to reduce the risk of infection in these vulnerable babies? And I bring this up because part of our study was looking at hand hygiene compliance for these moments. And so the other piece to keep in mind is moment 4 when we present some of our results. And moment 4 is when you're leaving this environment and you doff those gloves. So that would be moment 4, glove compliance. And you also, as you doff those gloves and leave this 1A environment, you would be, again, performing hand hygiene to reduce the risk of transmission from neonate to neonate. So that's sort of a little bit of a primer on how we talk about hand hygiene in the NICU. So again, there have been other centers that have started to ask this question. So this was published in JAMA Peds, and it was a single center RCT on extreme prems. They had a 45-bed unit similar to ours, with basically 7 pods or rooms where the neonates are sort of split between. Similar levels of acuity and similar infection control bundles in place. They randomized their 124 patients and in the end had 60 analyzable in each arm. And it was a longer study, it was over 30 months. Their outcome was a composite outcome. So it included episodes of bloodstream infection, urinary tract infection, as well as meningitis or CSF infection, and they included neck. And we'll talk a little bit more about neck towards the latter part of this slide. What they showed was that there was actually no difference in late onset infection. They showed a signal in a reduction in gram-positive infections, which is interesting because we do know that gram-positives can be sort of pathogenic and be on healthcare workers' hands, and potential transmission could be occurring through that route. They also showed a possible reduction in central line-associated bloodstream infections as well, 3 versus sort of 9 per 1,000 line days. So what can we learn from this study? Well, there were some limitations. It was unblinded, and there was obviously risk of contamination of their intervention. If a nurse is looking after one infant at one bedside who's randomized to one arm, and the neighboring infant they're caring for is randomized to another, you can see that practices might start to get contaminated across the board. Of course, they also individually randomized neonates and had to consent neonates. And so there may have been bias as to who entered the study. Their sample size calculation, unfortunately, was based on a much higher incidence than they saw. And so they were underpowered truly to see a difference. And because they included neck in their primary outcome, which has various mechanisms for its pathophysiology, it may or may not have sort of diluted the potential signal. And the other challenge, which we'll speak to in our study as well, was compliance was somewhat poor. They only monitored hand hygiene compliance, not glove compliance, and overall it was 79%. So this still sort of indicated the need for a pragmatic multi-center cluster trial with the appropriate outcome and sample size calculation with good compliance data to truly answer the question. So our next steps was to survey over 300 NICUs in Canada and the US to understand their current gloving practices. And what we found was 10% of sites were already using glove-based care. 57% of all respondents said that there was not enough evidence for a practice change already at this point in time, and 73% of respondents felt this merited further study. And interestingly, 96 respondents were interested in participating in a future multi-center trial on this question. So that sort of led us into the idea that this is worth sort of pursuing. And so our study primary objective was to understand if glove-based care decreases the risk of late onset culture-positive infection in NICU infants compared to standard hand hygiene alone. Our secondary objectives included looking at, does glove-based care reduce culture-negative episodes of infection, neck, all-cause mortality, length of stay, risk of colonization with AROs, and change in the gut microbiome and AMR genes in these infants. But we'll talk about how sort of that part of the study got a little bit shifted in terms of its analysis. So this, of course, was just a pilot study, just proof of concept, a single NICU cluster crossover study. So what this meant was the whole unit would go into the intervention arm or the control arm and cross over based on a randomization with a 2-week washout period in between. And the pilot was to last 6 months for each arm. We had, of course, being a pilot, feasibility outcomes that we were really trying to explore. So we wanted to see that would we actually get more than 90% of enrollment of the neonates, i.e. would people not opt out of our study? Was there more than 90% compliance among staff with the study arm? And can we get more data to inform a future sample size calculation and verify what we expect to be an appropriate event rate of late onset infection? So who was included? Basically anyone that stayed in our NICU for more than 2 days, because then they were eligible for an event of late onset infection. And we excluded anyone or their time period, but they may have needed isolation for other reasons, such as being colonized with an ARO. They're put in precautions which involve gloves. And so that data was censored from our analysis. In terms of the results, so I'll speak to our feasibility results first and some of the challenges we encountered. I'll speak a little bit to our infection analysis. Again, we're not powered to show a difference here, but it was sort of an interesting exploratory analysis to do. And as we talk, we'll see some of the lessons that we learned. So again, this was our pilot trial, and essentially we started in June 2017. We were randomized to the control or standard care arm first. We had our 2-week washout period after our 6 months, and then we went into glove-based care. And this is just to sort of show if any infant was put in isolation for gloves and gowns for various reasons, that data was censored for that time period. In terms of our numbers, we had over 1,000 infants admitted to the NICU. We had to exclude about 255 because of short lengths of stay. We had 390 in our control arm and 360 in our glove care arm after our washout. So to speak to some of our feasibility results, we wanted to achieve more than 90% of infants recruited, and in fact, nobody opted out. So that was sort of reassuring that we can proceed with a waiver of consent for a future multi-center trial. What event rate did we achieve? So we had 10% of neonates or 75 events of late onset infection occur. So that is comparable with what's known and seen in the literature. So that was also reassuring. We also wanted to look at sort of what type of resources would we require should we move on to a multi-center trial. And so we had 1 FTE research coordinator, and we needed significant extra data entry support. And I'll speak to the team that ended up coming together to get this study done. And that team needed to do sort of about 5 to 30 minutes to enter the data for a single LOI. And so sort of significant data entry requirements. We also looked at how long it took post-entry to adjudicate all the events. And again, it averaged out to 20 minutes per case. And we also sort of validated some of the data that was collected, and there was actual exclusion of a significant number of events. And so we do recognize we need to sort of tweak our data entry protocol to make sure that we're optimizing the quality of the data. So this is another busy slide, but I'll sort of simmer it down to key points. So one of our main feasibility challenges was getting good compliance data and achieving good compliance among hand hygiene and glove compliance. And this, of course, is a challenge of any sort of behavioral intervention. But sort of these first 4 columns speak to the fact that hand hygiene was significantly higher in the standard care arm compared to the glove care arm. And some of the reasons we hypothesized for this was that maybe people were starting to think that gloves replaced the need for hand hygiene and weren't doing the appropriate hand hygiene before donning gloves. Or perhaps requiring people to add gloves into their sort of routine care practices was sort of another kind of cognitive load that really threw people off, and then they sort of mixed up their algorithm of when they would wash their hands. The next sort of column really speaks to the fact that compliance with gloving was also suboptimal. You saw in the previous study, they achieved 79% compliance. We had about 66% for that moment 1 and 83% for doffing or moment 4. So really sort of suboptimal, below what we were aiming for in terms of our targets. And another sort of interesting thing to look at was, were people in the glove care arm at least either better or worse with their hand hygiene or their gloving? And in fact, people were actually better with hand hygiene before donning gloves than they were with gloving altogether. And so what this speaks to is the fact that behavior change is really hard, and trying to roll out a behavior change that impacts every care provider in the NICU is a bit of a difficult intervention to roll out. And we know behavior change is complicated and complex because you have to disrupt current habits and simultaneously foster new and possibly unfamiliar actions. And so it takes people like 2 months to add a glass of water to their day to become a consistent habitual behavior. So I think for a future trial, we really need to have sort of behavioral scientists play into how to optimize this aspect. Otherwise, we won't be able to answer the question effectively. So with that said, we'll talk a little bit about the infection results. So this is a busy slide, but the point of this is to say that really gender, where the babies were admitted from, GBS status in the mom, all these baseline characteristics, mode of delivery, rupture of membranes, antibiotics at delivery, indication for the antibiotics at delivery, and prenatal steroid use in premature infants, really there wasn't a difference across arms. So our randomization, if you will, did seem to work. We saw similar patients in both arms. What about specific risk factors we know for infection in both arms? How different or similar were those? And I'll just highlight sort of where we saw some differences. Interestingly, in the standard arm, there was a slight increase in the neonatal steroid requirements. So that's used for sort of pulmonary development. And there was what came out to be a statistical difference there. But the big piece here is probiotics, and you'll note that there was a significant difference here, and that's because there was a change in policy as it pertained to probiotic use in the NICU, and it rolled out just as we were starting our glove care arm. But more on that towards the end of my slide set. So let's just talk about the exploratory analysis we have around infection data. So all in all, there was no significant difference in the rates of sterile site infection across both arms. Similarly, there was no difference in gram-positive versus gram-negative infections, which was a signal that the other RCT had seen. And then when we talk about non-sterile site infections, so sort of a secondary outcome of interest, there wasn't a significant difference, but things like single positive coag-negative staph blood cultures that end up getting treated, or things called culture-negative sepsis, which are sort of subjective type infections, if you will, there seemed to be a higher rate among the glove care arm. When we looked at the rate of late onset infection per 1,000 patient days, there was a significant higher rate in the glove care arm compared to the standard arm. And this was when adjusted for known risk factors for infection, gestational age, birth weight, central lines, ventilation, and TPN. So we also wanted to look at what about possibly the mechanism that gloves might reduce the risk is in that early period, and perhaps we can delay their first severe episode of infection by adding gloves in that early windows of time where they're very extremely premature and first in the NICU when they have the most lines and tubes in place. And so we did see that the first episode of late onset infection, in yellow here, within the first 20 days of the NICU stay was significantly lower in the glove care arm. And this really sort of shows a little bit more clearly if you look at our survival curve, where in red here, the glove care arm is actually higher, i.e. we had a delay to first episode infection in the first few weeks, or the first 20 days of the stay, compared to the standard arm. Now that doesn't sort of last, that effect is lost over time. And it may be because sort of practices start to wane as an infant gets older and perhaps hand hygiene falls off. It's hard to sort of understand why this occurs, but we do recognize reducing that risk in that first window may be sort of a significant way to reduce negative outcomes for these neonates. So what can we say about all this? Do gloves prevent infection in the NICU? Well, in our small pilot study, we can't really say yes or no. Our data suggests there's likely an ability to delay the first infection in the highest risk neonates, which is their highest risk window for severe outcomes. The intervention itself, though, requires significant behavior change, and compliance is challenging to measure and truly implement. Our pilot confirms that a larger study will be needed to answer this question. We are now more clear on how to design a future study, and after having run this pilot, we have a better estimate on the sample size that will be required to see a difference and how to effectively and efficiently collect the data, so we don't need as large of a team. And I did want to sort of call out a huge thanks to the team that got involved, and specifically Kara Sang, a previous postdoc at the MacArthur Lab, as well as Jimmy who has helped significantly with a lot of the analysis. But we did have an army of volunteers that were mostly from the MacArthur Lab that really stepped in to help us with our data here. So now let's shift gears and talk a little bit about this whole wrench that got thrown into our study. So probiotics, what's the deal with that in the NICU? So we know necrotizing enterocolitis is a serious issue for premature infants in our NICU. And essentially, it's a syndrome where there's inflammation and bacterial invasion of the bowel wall in premature infants. It's very common in very low birth weight infants, but we see it in 1% of all neonates. The risk factors for it are if there's an ischemic gut injury, so something differently that occurs at delivery, or potentially an issue with the heart where you're not having good perfusion to the gut, or if there's rapid advancement of feeds, that can also precipitate this. There may be some degree of genetic factors as to why some neonates get NEC as opposed to others, but there's likely a big component of gut dysbiosis that's at play here. And the thought is that probiotics may modulate the microbiome and reduce the risk of NEC. So now we have to sort of give a primer on the microbiome, though I'm sure this audience is well familiar. The microbiome is the sum of all microbial life living on and in the human body. It's hundreds of trillions of organisms, and it actually is 10 times the number of cells in the human body. But most of that microbiome is sitting in that distal ileum and the colon, where we actually see NEC. Metabolic, nutritional, and immunological effects, the microbiome has a significant impact on all of these sort of pathways in the host. And the microbiome is not static, and it evolves sort of over time, and it may actually start to develop in utero. So there is vertical transmission that occurs from the maternal microbiota, and some of that may be occurring via the placenta in utero, as well as a significant shift that occurs at birth. We know that there are host factors as to how the microbiome may evolve, including the adaptive and the innate immune system. And of course, there are external factors that play heavily into how the microbiome will develop, including dietary exposures, antibiotic exposures, environments like the NICU, and illness impact the developing microbiome. So in terms of the neonate, we know mode of delivery is a huge factor. So vaginally delivered infants have much more lactobacillus and Prevotella present, whereas infants born by C-section actually end up with more epidermal species, things like Clostridium, Staph, Propionibacterium, Cranibacterium, and much less anaerobes are present in these infants. And it's interesting because the difference in diversity and function of the microbiome that may occur at birth can persist for months after birth and even up to years. We know that the first week of life is when gut colonization really shifts, and that can have significant impacts on the future microbiome. And we also know that even a term neonate's microbiome matures over a year of age, and it doesn't sort of become into its adult form until about 3 years of age. So when we talk about the premature infant, which is much more of who comes into the NICU compared to the term infant, we know they have reduced diversity in their microbiome. It's much less stable. They have a much more immature immune system and gut, and they have a much higher risk for pathogenic organisms to get in and possibly precipitate episodes of NEC. In terms of what types of bacteria we see there, there's many more facultative anaerobes, which are much more likely to be pathogenic, things like Enterococcus, Enterobacter, and Staphylococci, whereas term infants have much more sort of balanced anaerobes, which are much less likely to end up causing disease or precipitate NEC. In terms of the evidence for probiotics in NEC, there's actually been large RCTs, and we now have multiple meta-analyses. So very few areas of neonatal research are this sort of rigorous, but there has been very clear understanding around probiotics really competing out against pathogenic bacteria, generating the right metabolites signaling to sort of reduce risks of pathogenic organisms, decreasing inflammation, tightening up gut barriers, optimizing immune cell regulation. And all of this is likely why we see such a significant favoring of probiotics, or a positive impact of probiotics on reducing the risk of NEC. Interestingly, there are some smaller data that also suggest a potential reduction in death and length of stay, though I think the jury's a little bit more out on that. And so with that sort of overwhelming amount of data, there really was a shift in our NICU that occurred, of course, right in the middle of our study, where essentially a policy rolled out that all infants less than 34 weeks of gestational age or with a birth weight less than 2 kilos were to receive probiotics. And of course, if you had congenital GI issues or immune deficiencies or calcium protein allergy, other reasons why you were not to be receiving probiotics. Of course, giving live bacteria to someone with confirmed sepsis probably isn't a great idea. But for the most part, many of our neonates were getting probiotics. And what you might ask is, in the probiotic that we are using, it's sort of multiple strains of bifidobacterium and a lactobacillus. So our initial plan was to look at how does glove-based care affect the neonatal microbiome and the resistome in the NICU, but that changed once it became clear that likely probiotics are going to alter that microbiome in a much more significant way than what we would see from gloves alone. So our sort of sub-study in the glove-based care study was to get an admission swab of feces and a discharge swab and see how the microbiome evolves through the NICU stay, as well as look at some of the AMR genes that are present. And so we did have 25 babies that had to be consented because there was sort of swabs being collected at admission and discharge in our standard care arm, and we had 21 babies in the other arm. And we did match based on vaginal versus C-section delivery and gestational age, knowing what we know about the microbiome. And of course, as I mentioned, the glove care arm, really, it became a probiotic look as opposed to a glove care look. And another wrench, the COVID pandemic hit. And so really the analysis is still sort of underway. And I think the MacArthur Lab is still sort of hoping to get this done sooner than later. And hopefully maybe we'll come back with more data from our specific study. But with that, I'll hand over to Allison to speak to some of the interesting stuff happening, looking at the microbiome and the AMR resistome in the NICU in their study, which sort of rolled out at around a similar time. So take it away, Allison.
So we'll shift gears a little bit, still within the NICU realm, looking at preterm infants and probiotics, but this cohort that I was looking at did not fall under that glove-based care study, as Sarah had mentioned. And so Sarah did a great job introducing all the different disadvantages that preterm infants have in terms of their development. And I just wanted to highlight kind of two things that pertain to what I'm going to be speaking to you about today, is that preterm infants usually spend an extended amount of time in the NICU, and that this puts them at greater risk of exposure to pathogens. And so these bacteria exist in the environment. And as Sarah mentioned, there's different interventions to try and prevent them being transferred to preterm infants. And one of the things that we're most concerned about is the antimicrobial resistance potential of these pathogens. And so another issue with preterm infants is that almost initially they're given antibiotics such as ampicillin and gentamicin, and then depending on how they progress, they might be given a whole variety of antibiotics. And so we are really concerned about how these antibiotics might be further selecting for either colonization or persistence of those multi-drug resistant clinical pathogens in their gut microbiome or other microbiome environments. And so, as Sarah mentioned, probiotics have kind of come into play in terms of potentially reducing infections, including necrotizing enterocolitis, but also from another side of the coin is looking at how they might help with the development of the gut microbiome. And so we were interested in seeing if we could monitor how probiotics might be impacting the microbiome as well as antimicrobial resistance. And our hypothesis was that probiotics would be able to reduce antibiotic resistance genes associated with pathogenic bacteria in the preterm gut. So in order to study this kind of question, we were collaborating with Drs. Eileen Hutton and Dr. Catherine Morrison here, and they had initiated two large cohort studies known as the Baby and Me and the Baby and Pre-Me study. And so this was done many years ago, but essentially they were recruiting mothers that were tested for group B streptococcus and either given intrapartum antibiotics or not. And they were following these babies up to 3 years of age, and they did stool sampling, they looked at different growth parameters, and really they were interested in understanding how the microbiome developed over time and whether this early life exposure had any kind of effect. And so there's a paper that came out looking at the impact of that intrapartum antibiotic. And more recently, they started recruiting preterm infants into this cohort with the same kind of idea of understanding these early life exposures and how they impact the development of the microbiome. And as Sarah mentioned, there was this change in the NICU where certain preterm infants were now given probiotics. And so a study came out from a PhD student, Efra Youssef, that looked at the persistence of these probiotic strains. So the same probiotic that Sarah mentioned, we could see persistence of that bifidobacterium in these infants up to 5 months of age. So looking at these two cohorts, we had a good opportunity to see how probiotics might be impacting the development of the microbiome and antibiotic resistance within the microbiome. So for our study, we looked at 3 different cohorts of infants. So we have preterm infants that were given antibiotics, but were not part of that probiotic change in the NICU. And then we have preterm infants that were also given antibiotics, but in this case, they were given that probiotic while they were in hospital up to discharge. And some infants continued probiotics after discharge. And so for these two preterm cohorts, we have samples, stool samples that were taken while they were in hospital, and then on follow-up visits up to 5 months corrected age. From the baby and me cohort, we also have some full-term infants. These infants did not receive antibiotics nor probiotics, and we have one time point for these infants. And so that's a 10-day sample, which can be, we matched to the in-hospital sample. So for the preterm infants, the in-hospital sample was taken around their term corrected age. So the full-term infants are slightly older, but it was the best comparator we had to see what differences might exist between preterm infants and full-term infants at this early age. And so to try and understand what's going on with the microbiome, we often turn to genetics and DNA sequencing. And so we can take a stool sample, which is representative of the gut microbiome, the best representation that we can get, and we can extract all the DNA from that sample. And so this is what we often refer to as the metagenome. And this is a complement of DNA from the host, from bacteria, from fungi, from viruses, anything that's within that stool sample. And if we want to try and profile the microbiome or the bacterial diversity within that sample, we can do something called amplicon sequencing or 16S rRNA sequencing. And this is one of the better proxies that we have for understanding the diversity of the microbiome, apart from just sequencing everything that's there. So this was done, as I mentioned, by a PhD student, Efra Youssef, and kind of the big result that you get from it is looking at what different bacteria might exist within your samples. And so the image we get from it is not the nicest representation, but I'll walk you through what I'm trying to show you here. So I have our different time points that we sampled at. So when the babies were in hospital and then up to 5 months of age or 5 months corrected age. And along the bottom, I have our different infant samples that we had. So NS are the infants that didn't get probiotics, the preterms that didn't get probiotics. PS are the preterm infants that did get probiotics. And then FT are the full-term infants that didn't have probiotics and didn't have antibiotics. And so, as I mentioned, I only have one time point for those full-term infants. And a few things I want to point out to you here are these blue colored bars and these pink colored or salmon colored bars. And so what this is representing is the relative abundance of a particular signal within the microbiome. And this signal is associated with Clostridiaceae and Enterobacteriaceae. So I believe Sarah mentioned kind of the development of the microbiome of preterm infants, and these types of bacteria usually show up quite early. And what we see is that they're usually only associated with infants that didn't get probiotics, and that they do, some of them tend to persist up to 5 months of age in the preterms without probiotics. Another thing I want to point out here is this kind of purple colored bar, and that is a Bifidobacteriaceae. So I can't specifically say if it's the, well, Efra was able to show that it is one of the strains that was given in the probiotics. So you can see that that purple bar tends to be more dominant in preterm infants that were given the probiotics, suggesting, as we hoped, that the probiotic would seed or colonize the gut microbiome, and that we see that almost predominantly in the babies that were given probiotics. It does show up a little bit in the non-supplemented preterms, and that's just kind of the development of the microbiome over time. So really what we're seeing is that probiotics are effective in colonizing the gut microbiome, and that babies that didn't get probiotics, their microbiome seems to be more dominated by Enterobacteriaceae and Clostridiaceae. And Enterobacteriaceae is kind of one of these groups of bacteria that we're a bit more concerned about because it often contains things like E. coli and Klebsiella pneumoniae. And those we know are often associated with antibiotic resistance and potentially causing nosocomial infections. So that's kind of what it looks like from the microbiome point of view. When we want to study antibiotic resistance, we often refer to this as the resistome. And it's a little bit different than studying the microbial diversity because antibiotic resistance genes only represent a very small proportion of the total metagenome. So before I was showing you all this different DNA, but the resistome is just a subset of that DNA. And so through various efforts and research, we've determined that over 4,000 genes can be implicated with antibiotic resistance. So bacteria have really acquired this kind of arsenal of potential to protect themselves from antibiotic exposure. And one of the bigger concerns also is the fact that these genes are quite mobilizable. So they can be captured on plasmids, which can then spread between different bacteria, potentially spread between different environments. And so this is kind of one of the major concerns with antibiotic resistance to date. In terms of what we know about antibiotic resistance from the microbiological side of view, we can zoom in on a bacterial cell and we can look at their genetic material and we can pinpoint different genes within the chromosome, or as I mentioned, on plasmids that are associated with antibiotic resistance. And so these genes encode proteins, which serve some sort of function to protect the bacteria from antibiotic action. And this can occur through producing these membrane spanning pumps that expel antibiotics from the bacterial cell. You can have different modifications on the cell membrane that prevents antibiotics from entering into the cell. One of the largest classes or families of antimicrobial resistance are the antibiotic inactivation enzymes. And so beta-lactamases are a key example of this, where they've really expanded. There's so many different varieties. They're quite widespread among different pathogens. So really that's kind of the key player in this antibiotic inactivation mechanism of resistance. You can also have different enzymes or proteins that actually modify the target of an antibiotic. So many antibiotics target essential processes like transcription, translation within bacteria. And so that these processes all involve different machinery and proteins. And so if an antibiotic can no longer interact with that protein, then the bacteria is protected. So you can have modification of the target. You can also have modification of the antibiotic itself. So different enzymes can add different chemical groups to the actual antibiotic scaffold so that it can no longer intimately interact with that protein partner. And then finally, this is often seen in kind of chronic infections where a subpopulation of bacteria have a mutation, which is beneficial in terms of protecting them from antibiotics. And so if that mutation is selected for, then that population of bacteria can thrive. So there's just a variety of different kind of higher level classifications of how bacteria can be resistant to antibiotics. And it is quite a complex phenomenon to study. And so how can we actually try and understand what's going on at the resistome level in our babies? The one thing we can do is, as I mentioned, extract all the DNA, look at everything that's there. And this method is kind of a brute force method where you just sequence everything, and it's referred to as shotgun sequencing. So what I'm trying to show you here is that the black lines represent any DNA that's kind of background or host related, and that these kind of colored lines are antimicrobial resistance genes or DNA that's associated with AMR. And in a typical shotgun experiment, we find that about less than 1% of the data we obtain back is associated with antimicrobial resistance. And so when you're dealing with a large cohort study or longitudinal sampling, this really becomes expensive and cumbersome to sequence everything that's there in every sample if we're only interested in less than 1% of that data. Obviously, the rest of the data might be interesting and give us extra information, but if you can't afford this type of experiment, then perhaps you want to try a different approach. And so what we've been doing throughout my PhD is this more targeted sequencing approach, which relies on designing probes that are specific to antimicrobial resistance genes. And this is often referred to as fishing, or pulling out, looking for the needles in the haystack out of a larger complex metagenomic sample. You can pull out and specifically enrich DNA that's associated with antimicrobial resistance. And what we've seen and others have also seen is that what you get back is this kind of enriched pool where greater than 50% of the data that you're sequencing can be traced back to antimicrobial resistance. And so when I first joined McMaster, this project had already been initiated by my supervisor, Dr. Jerry Wright, and Drs. Andrew MacArthur and Hendrik Poinar. And Brian from Dr. MacArthur's lab was tasked with designing this probe set. So as I mentioned, these little sequences are complementary to what you're trying to target. And so the way that Brian did this was he took every single antimicrobial resistance gene that we knew about at the time and tiled across these short oligonucleotide probes against every single resistance gene. And this was done in combination or in collaboration with a company out of Ann Arbor, Michigan, known as Daysell Arbor Biosciences, and their technology is referred to as MyBaits. This technology is available through different suppliers, and essentially you can design your probes for anything that you could imagine if you know the DNA sequence for. So how this method works, just a bit of background into it. Essentially, as I mentioned, you can start with a DNA extract from any sample that you're interested in. In our case, we have all those preterm and full-term stool samples. And so you take that DNA, you prepare it as you would for metagenomic shotgun sequencing. And as I mentioned, we're expecting that our target is quite minute in the total DNA complement. You take that, you mix it with your probes. These are really efficient kind of fish hooks for your DNA of interest, and you can wash away all your background unwanted DNA. And hopefully what you're left with at the end is an enriched pool of your target. And so in this case, we're hoping that we have all our antimicrobial resistance genes from our starting samples. And so I did this approach for all of our preterm infant samples and our full-term infant samples, and I wanted to compare what antimicrobial resistance genes are there, what are they doing, and do I see a difference? So the first thing I looked at was the number of antimicrobial resistance genes, and I wanted to see if there's any difference kind of in terms of resistance gene load between our infant groups. And so this is just the first time point that we had samples for. So early on in life, when the preterm infants were in hospital and the full-term infants were 10 days of age. And what I'm showing you here is that every single point represents an individual preterm or full-term infant gut microbiome or gut resistome. And what I was counting was how many resistance genes do I see in one infant that I don't see in either of the other infant groups. And so you can see that in the non-probiotic groups, so the preterms that didn't get probiotics, there does seem to be a greater diversity of resistance genes that we don't see in either of the other two groups. And now this is just the number of resistance genes, but it doesn't really tell me if those are resistance genes that I should care about or be concerned about. And so, as I mentioned, you can break down resistance into different mechanisms, different classes. And this is what I'm doing here when I'm looking at what are those resistance gene families. And so these names might not mean much to you, but to someone who's studying resistance, you can get a lot of information from just a name of a resistance gene family, like what antibiotic it confers resistance to, how it confers resistance. And you start to recognize some of these names after a while. But what I'm showing you here is that anything that's colored in teal is if I saw that resistance gene family in one infant in that cohort. And you can see that there's definitely certain ones that are exclusively found in infants that did not get probiotics compared to the probiotic supplemented and the full-term infants. And so just wanted to point out kind of these blocks of genes here. Anything that starts with an A, kind of like AAC or ANT or APH. Those are aminoglycoside modifying enzymes or resistance genes. And then these BLAs, those are your beta-lactamases. And so not surprisingly, all preterm infants are given Ampicillin and gentamicin. We would expect to see these kinds of genes in our preterm infant gut microbiomes. So overall, what I'm seeing is just at this early age already, there seems to be a stark difference in the types of resistance genes that we're seeing in probiotic supplemented versus non-probiotic supplemented preterms, and that there does seem to be this reduced diversity of what we refer to as the antibiotic inactivation genes, or in the case of the aminoglycosides, the antibiotic modifying genes. So what's happening longitudinally? Are we seeing a difference at 5 months of age, which I think is really interesting to look at? And so I can repeat the same kind of analysis, looking at how many genes I see in one group of infant versus the other. And there's a general trend. So this is from when the babies were quite young, while they were in hospital, up to 5 months corrected age. And in black, we have preterms that didn't get probiotics. In kind of pink, we have preterms that did get probiotics. And you can see that there just overall seems to be an increasing diversity of antimicrobial resistance genes that are uniquely associated with non-probiotic supplemented preterm infants. And again, we can look at what those resistance gene families are. And so what I'm showing you here is that kind of presence absence plot again. So on the left-hand side, we have preterms that didn't get probiotics. On the right, we have those that did get probiotics. And then I have that different, or those different time points that we sampled at. And so you can see that there are certain resistance gene families that are uniquely associated with babies that didn't get probiotics, and that these tend to persist up to 5 months of age. So really, I'm getting this picture that there's an increased resistome in babies that didn't get probiotics, and that we could almost say that probiotics are reducing the diversity of antimicrobial resistance genes in the preterm infant gut up to 5 months of age. And so this is just, looking at it from a gene perspective, but these genes are encoded within bacteria. They might be encoded within plasmids. So really, can I say by just looking at the gene, what particular bacteria might be carrying that resistance gene? And so there's some analysis that I can try to do to say what bacterial families might be carrying those resistance genes, trying to relate it back to the microbiome. So this, what I'm showing you here is kind of a association plot or a connection graph. If I saw a resistance gene, which are these circles, was I able to trace it back to a particular bacterial family, which are these squares. And so this is for the preterms that didn't get probiotics and kind of an overall look across all time points. And you can see that there's many different resistance genes. The purple ones are those that confer resistance to beta-lactams, and the green ones are those that confer resistance to aminoglycosides, because those seem to be the most predominant ones that I was seeing in the gut resistome of these preterms. And so this is the picture for non-probiotic supplemented. For the probiotic supplemented, it's a much smaller picture. There's fewer AMR gene families, fewer bacterial families. I do want to point out that we do see resistance genes associated with Bifidobacteriaceae. This is not quite alarming. We know that these bacteria would be intrinsically resistant to antibiotics, but it's a good kind of confirmation that these babies had Bifidobacteriaceae because we're able to pull out antimicrobial resistance genes associated with Bifidobacteriaceae, but we don't necessarily see those as predominant in the non-probiotic supplemented. I do want to hone in on this one interaction here between Enterobacteriaceae and this beta-lactamase called SHV beta-lactamase, because it was an interesting case study in the preterms that didn't get probiotics. And so diving deeper, kind of a gene level view of these resistance genes, this is how I see the data. So every little purple region here represents that resistance gene, and then anything beyond that is kind of, if I was able to capture data beyond that, because this is not metagenomics, you're not getting these kind of long pieces of DNA, you're getting shorter fragments. And so you're not able to see a full view of where that gene is, whether it's on a chromosome or on a plasmid, but you do get some surrounding information, which you can look at and kind of get other ideas of where that gene might be. And so what I'm showing you here is every time I saw this resistance gene and the baby that I saw it in and the time point that I saw it in. So you can see that it's pretty much predominant in babies that didn't get probiotics, and we can see it while they're in hospital and then up to 5 months of age. So this resistance gene, and by proxy, perhaps its bacterial host, is persisting in the gut microbiome of these preterm infants. And so SHV beta-lactamases are well known to be associated with Klebsiella pneumoniae. They can also be found on mobile genetic elements. I can't quite say if that's the case for how I see it in the gut microbiome of these babies. And I believe the exposure likely happened in hospital, and the microbiome was just primed for this Klebsiella pneumoniae, which is part of Enterobacteriaceae, to kind of persist in the gut microbiome of these infants. And so just some quick concluding remarks here. I hope that I have kind of highlighted how preterm infants are quite exposed to potential multi-drug resistant clinical pathogens in the NICU, and how this can have various disadvantages for them as terms of how they're developing, and perhaps they could even be colonizing the gut microbiome of these infants if the conditions are right. Preterm infants also get are exposed to many different antibiotics in the NICU. And what we're concerned about, of course, is that selection for multi-drug resistant clinical pathogens in the microbiome. Hopefully, we've shown you today that probiotics seem to have some benefit in terms of reducing necrotizing enterocolitis, but also in hoping to promote the development of the gut microbiome. And what I think still needs to be teased out a bit more is whether they're having a direct impact on potential multi-drug resistant clinical pathogens from colonizing the gut microbiome of these babies or not. And I did want to just kind of prime for what this might mean for the later life for these preterm infants. Because we only study their microbiome up to 5 months of age, we don't know what's happening beyond that, but we do have some information in terms of whether these babies were given antibiotics later on in life. And so by 2 years of age, we know that about 70% of preterms that did not get probiotics received antibiotics later on, compared to no of the probiotic supplemented infants. So because we already know that these babies that weren't given probiotics have kind of enriched resistome, it is quite concerning that they are receiving antibiotics later on in life, and those antibiotics might be further selecting for those multi-drug resistant clinical pathogens that have now colonized the gut microbiome of these infants. And so I don't know if Sarah wants to chime in here once I finish kind of the take-home points, but what Sarah showed us today is that infection prevention, there's many different preventions that you can introduce, and it is an ongoing challenge in the NICU, and that antibiotics might have significant long-term impacts on neonates. And I think what was really interesting is in Sarah's case, there were these confounding factors that came into play, especially in terms of the probiotic exposure. But at least they were able to show that gloves may delay the onset of infection in NICU infants. What my study has shown is that preterm infants, well, we already knew they have a diverse set of antimicrobial resistance genes in their gut microbiome, and that at least in our longitudinal study, we do see that these genes persist up to 5 months of age. And then finally, some of the offsets of this probiotic change in the NICU is that there might be some impact in terms of helping with the development of the gut microbiome and reducing persistence of AMR and these kind of pathogen-associated resistance genes in the gut microbiome of preterm infants. And so I do have a lot of people to thank. Obviously, the 2 cohort studies led by Dr. Eileen Hutton and Catherine Morrison, all the patients that were involved in these studies and the midwives that recruited them. Dr. Jennifer Stearns was kind of the lead PI on this preterm microbiome study, and Efra Yusuf was very helpful, as well as Amos from the MacArthur Lab helped out with the bioinformatics side of things, my committee, the sequencing facility, and all our funding. And I think Sarah has some thanks as well.
Yeah, just briefly, of course, our NICU patients that enrolled and allowed us to do this data collection, the Wright Lab, MacArthur Lab that are working on some of the next steps. And of course, the large team of volunteers, students that assisted, and including the Wright Lab and Allison's work and the Pointer Lab, and then our funding sources, including the NIF, the IIDR Research Institute, and the Boris Foundation.
Thank you so much, ladies. I'll give you a clap in proxy of the rest of the audience. So while we're waiting for questions to appear, I have one quick question. And I'm curious because, Sarah, you mentioned that this probiotic supplementation started in 2017, which is, that's now 5 years ago, and the science has progressed. So is the selection of the probiotics different now than what you were using then, because we know more about what to give the babies?
Interesting question. So there are very few sort of probiotics that have been sort of studied and validated for use in even premature infants. So there's really a narrow spectrum of options, and then sort of the process to get it through like FDA and then formulary approval at our site. There's not a lot of movement, I think, happening right now, but not to say that I think the research that's being done here and elsewhere may inform that and be more targeted. For now, it's a bit of a well, this has been studied. And that's another piece to add. A lot of the RCTs used a variety of probiotics. And so some of the people that question, truly what is the impact and where is the effect and what probiotic is making the difference, there's a lot of diversity in the studies, and we sort of just lumped them all together for much of the analysis.
Okay, thanks. So there's a question from Jake. For Dr. Khan, how did the study measure behaviors, hand hygiene, gloves, et cetera, when assessing feasibility? And if it was a participant-reported measure, how did the study address behavior reporting bias?
Great question, Jake. So this is like the crux and the challenge with our study. The trouble with hand hygiene compliance monitoring unto itself is that for the most part, it's external auditing. And so there's a significant Hawthorne effect. We know the validity of that data is skewed, right? Things like recording or having observance that's not obvious would be ideal, but isn't typically ethics approved. And so we're stuck with external auditors. We did randomize their presence there, i.e. like day shift and night shift, really tried to get the breadth of care in the NICU. It was sort of 3 hours per week. So that was how our compliance data is collected. It's a small subset of what actually happens in the NICU, and it does suffer from Hawthorne effect. One of the reviewers of our paper mentioned, could you have done some of these other more intensive measures? They're resource intensive and often wouldn't get through ethics. I forgot the second part of the question. I don't know if it's disappeared.
Bias in behavior reporting.
Yeah, so we allowed for self-report. We allowed for self-report in terms of like if people knew they missed gloving, i.e. like they were unable because of acuity to don the gloves before intervening. We did ask them to report that because we wanted to understand what are barriers for gloving. It was minimally done in terms of recording, because self-reporting we know suffers from its own biases. So these are limitations clearly for any type of intervention of this sort.
I feel like these are the kinds of studies where you actually need to have a monitor, because the people who are doing the intervention are so busy doing the intervention that, things may become a bit blurry in terms of reporting.
Agreed.
I have a quick question for Allison. So the microbiomes that you were looking at across the very, very young babies and then the little bit older preterms, I'm assuming all of those kids got antibiotics as a routine matter of course. And I just wondered how that impacted potential engraftment of the probiotics if they're getting antibiotics. So is there a timing allowance to make sure that you're not giving antibiotics and probiotics simultaneously?
Yeah, so I don't know the exact timing in terms of when antibiotics might have stopped and probiotics might have started. We do know that after probiotics started, none of those infants received antibiotics during that study period. So there is kind of that anecdotal evidence. Right. But yes, the antibiotic exposure is usually quite early on, I guess, as they're developing and kind of going through the process and maybe, getting sick or getting better, and depending on how they're progressing. So yeah, I don't have the exact, like, if there was a washout period necessarily.
Yeah, I was just wondering whether if that could partly explain the differences in your data, right? Because if the probiotic group is not getting antibiotics, the signal presumably for bacteria that have those genes would be lower because they're not being selected for.
Yeah. Yeah, so there was, like, I did try to control for all the different antibiotic exposures. It was quite varied across the board. They all did receive ampicillin, gentamicin, but then so many different antibiotics throughout kind of the first couple of weeks. And so in terms of that, yeah, I would say that probably, they received a similar amount of antibiotics prior to when the probiotics started.
Yeah, it's a pretty impressive effect, I have to say. Okay, we have one more quick question. I know we're at time. Dr. Smale wants to know if you can control for breastfeeding versus formula.
From my point of view, I think about 50%. I don't know if they were all breastfed or formula fed, but I do know, like, if they were weaned during the study period. So I don't know if Sarah can talk about how maybe they might want to look at controlling for that in their study.
Yeah, no, that's a great question. We didn't, and I think there is data to suggest that that those dietary influences significantly alter it. Most of the prems, if they're not receiving maternal breast milk, are donor breastfed. So I think didn't match on that parameter. We tried to sort of choose maybe what's a more significant impact in those early days. But I think it's a relevant point, and we can look at it sort of if we do have kids that receive more formula. But again, the vast majority of our prems do get EBM. Express breast milk.
Okay, well, we're at time now, so I just want to thank both of you again for your great talks. It was really an interesting morning. I learned a lot. So thank you again. And thanks to the audience. We will see you next month.